Hello, my name is Jacob Torres, and I am a PhD student and researcher in Dr. Adam Dolezal's lab at the University of Illinois' Entomology Department. Today, I will be telling you about our exciting new study monitoring factors that affect honeybee health, a slick new website and app being developed for beekeepers, and how you can get involved as an individual beekeeper and citizen scientist. Before I go further, I would like to give you some background information on who I am and what we do in the Dolezal Bee Research Lab. I am currently a PhD student at UIUC and have obtained a master's degree from Western Illinois University, where I studied bee gut microbiomes, and a bachelor's degree from Augustana College, where I had the pleasure of working on several different insect systems. In the Dolezal Bee Research Lab, we are primarily focused on understanding landscape-level predictors of bee health, which means we like to ask questions like, what beekeeping practices work in two distinct landscapes, and what features are associated with better, healthier, and more productive bees? As you can see in the figure to the right, honeybee health is not a simple problem to solve because of the many factors or features that not only affect honeybee health, but are also interacting with one another. Understanding how these complex interactions between stressors like disease, nutrition, and pesticide exposure impact honeybee health therefore requires a massive set of information that can only be obtained by sampling and collecting data from a large number of hives. But how do we determine what predicts a successful honeybee colony at the landscape level? To start to answer that question, let's quickly look at other studies that ask similar questions. In this study that was conducted in the UK, the researchers found that, generally, there was a lower percentage of protein found in the bee bread of hives located in areas that were agriculturally intensive compared to the bee bread of hives that were further removed from agriculturally intense land. There was also a similar relationship seen in the United States, specifically in the Dakotas. This may suggest that this pattern is one that can be observed throughout the globe. In this study conducted by Dr. Adam Dolezal while a postdoc at Iowa State University, beekeepers with hives located across an agricultural gradient ranging from 17% cultivated area to 91% cultivated area collected and sent a sample of live adult bees from multiple hives. Dr. Dolezal was then able to analyze the samples and record information associated with honeybee health, such as quantifying pat stores, counting mite loads, and identifying viruses present and their quantity. This information was collected before the hives overwintered, so you can imagine how you may be able to compare hive health before and after overwintering. One of the findings from this study was that landscape use had an impact on bee lipid stores. However, this effect disappeared in the presence of Varroa mites. As you can see, these landscape studies are all building a bigger and better picture. One of the problems biologists often face is that their study is only a snapshot of what is happening in specific areas from which they have sampled. This, however, does not allow us to understand the underlying mechanisms responsible for the patterns we see the way we might be able to if we were conducting a controlled experiment in a laboratory setting. For example, if I were to collect information from all the hives in the Chicagoland area, I would likely have a really good idea about what impacts colony success in that area. However, if someone from Southern Illinois asked me to give them recommendations in rehabilitating a hive of theirs, I probably would not base my recommendations on the pattern I saw in Chicago. This is because two different areas will have different levels of how each factor affects honeybee health. This gets ever more complicated considering that the same factor can have different overall effects on honeybee health when combined with other variables and interactions. In order to understand how these variable landscapes affect honeybee health, we must therefore ask questions like, what predicts colony success at the landscape level? And vice versa, what predicts colony problems at the landscape level? By collecting data from as many hives as possible, we can begin to see how certain landscapes and their variables may promote the spread of Varroa mites, viruses, and other diseases, while other landscapes may discourage their spread. Likewise, we can also begin to understand what landscapes provide bountiful forage for bees and which habitats may cause bees nutritional stress. By collecting information to answer these questions, we can also begin to see how widespread these trends we see really are, rather than just having local snapshots. One of the reasons why this study is so exciting are all the resources we have available to us in this state, as well as the characteristics our state exhibits. Illinois also spans 390 miles from its northern border to its southernmost tip. This allows for a wide variety of climates along a long north to south gradient that can impact honeybees in a variety of ways. One example of how this gradient of climates could impact bees is in the availability of forage. Hives that are kept towards the northern border of Illinois will likely experience a longer and harsher winter than hives kept towards the southern tip of Illinois. A longer and colder winter means that plants in the northern end of the state will not be in bloom until later in the year compared to plants that experience shorter and milder winters. This not only affects bees by affecting the types of flowering plants available as forage, but also by extending the period of nutritional stress endured by northern bees. Illinois also has a huge variation in population density across the state, with the majority of the population concentrated around the Chicagoland area and other smaller urban areas. This provides us multiple gradients as the landscape morphs from a highly urban area to suburbia and then to agriculturally intensive land or natural areas. As I mentioned earlier, this allows us to get a better understanding of the factors that impact bee health in specific areas, but also in the transition zones between those areas. This not only gives us a bigger picture and better understanding that can lead to improved bee management practices, but also allows us to narrow in on the effect an environmental variable has by comparing distinct landscapes with similar values for any given variable. The most exciting thing about Illinois, in my opinion, is the sheer number of hives available as potential data samples. There are over 24,000 colonies and 2,500 registered beekeepers across the state. I can only imagine the patterns that we might observe and be able to learn from if we could collect information from every single hive. An important reason why we need you to participate in the study as citizen scientists. Because of these great features, Illinois provides us with an excellent opportunity to understand how a wide variety of factors influence bee health and what beekeeping practices are effective across space and time. This can then lead to better management of honeybee hives for our region. We have all this collective knowledge about bees and beekeeping out there across the state, but we don't have any way to collect that information and start to see these big trends. So, how can we go about collecting this type of data? An exciting feature of this study is a new website and app that have been designed by Christina Grosinger and other bee researchers at Penn State with private beekeepers in mind, called beescape.org. The beescape.org website and app will allow you to register yourself as a beekeeper, upload data that you collect from your hives, and get a score of how your colony will likely fare for survival, how your hive's honey production compares to the region, the forage quality surrounding your apiary, and much more. You will be able to do this by finding your apiary on a map, selecting it, and seeing what landscape variables are directly impacting your location. Some of the variables that will be reported include a score of pesticide use or exposure in the area surrounding your apiary, a ranking of native bee habitat available around your apiary location, and the quality of forage around your apiary during different seasons of the year. All of this information is kept anonymously, so your personal information and name will never be shared with anyone, only the data collected from your hives. Access to this website and app is also free to all Illinois beekeepers. As you can imagine, this can directly impact the way we manage our bees, where we choose to start an apiary, as well as the regional decisions we make as we try to bolster native and honeybee populations across the globe. So, you want to get involved as a citizen scientist and help us collect data from your hives. That's fantastic. Now, what exactly do you have to do to participate? There are two levels of involvement that you can commit to as a citizen scientist: a light and a pro survey option. The light survey option only asks that you provide data on your management practices and hives in November and April, while the pro survey option involves a monthly survey. The monthly survey will be available after the 15th of each month. In order to participate in this study, you would have to randomly select hives in your apiary and inspect them once a month after the 15th of that month. There is a schedule on the project website listing the dates on which we would prefer you to inspect your hive. During this inspection, we are asking that you record certain information such as the size of your colony, Varroa mite counts, hive history, and the beekeeping practices that you employ. All of this information is entered into an online form through beescape.org, and the inspections are repeated once a month using the same hives as before. That's it. This expanded inspection shouldn't take too long for you to complete, with an average inspection lasting about 10 minutes. As for Varroa mite counts, there are several different approaches that you can take. As we do our inspections, we will be using a Varroa Easy Check, which is the same system state apiary inspectors used during their data collection last year. However, if you don't have an Easy Check and wouldn't like to invest in one, there is a way to do an alcohol wash using common household items. For the Varroa mite counts, we are asking that you take a random sample of bees from your hive with roughly about 300 bees. The Easy Check system has markings that give you an estimate of how many bees you are sampling, but a simple half-cup measuring cup filled with bees is roughly 300 bees. As you work through your hive, you can randomly select a brood frame and brush bees into a half-cup measuring cup until it is full. Once full, you can pour the half-cup of bees into a Tupperware container filled with ethanol or isopropyl alcohol and cap it. Vigorously shake the Tupperware container for 30 seconds to a minute to euthanize the bees and mites present and dislodge any mites that might be attached to a bee. The bees can then be poured onto a strainer, and the alcohol and Varroa mites will pass through, allowing you to count how many mites were present. As you can see in the uppermost figure, you may also modify two jars by attaching them together with a screen separating the two containers. This will allow you to shake the sample, strain the sample, and count the mites all with the same tool. If you prefer, you may also measure mite levels in your hive by doing a sugar roll, an ether roll, or by using a sticky board. It is important to remember that if you choose to use a sticky board, you must report the number of mites present over the amount of time that the board was in the hive. In conclusion, this study is not only exciting, but extremely important because it will allow us to see and start to understand large-scale patterns of Varroa mite buildup and hive strength. We can then compare these large-scale patterns with the season or time of year, beekeeping practices, and land use to begin to understand what factors impact our honeybee hives the most and make recommendations to increase the strength and productivity of our hives. This also builds a large database for tracking hives, which will allow beekeepers to have a better understanding of how their hive compares to those in the same region, and may lead to some beekeepers making adjustments to their beekeeping practices to encourage their hive to be healthier, stronger, and more productive. In future years, we could expand the data collection to include other pathogen measurements as well, and record other hive qualities which may allow us to not only see the spread of mites and viruses, but also be able to predict and manage their spread. We will also have a stronger understanding of how forage quality and availability impacts honeybees, and will be able to predict what types of landscapes are likely to put your bees in high levels of nutritional stress, and what landscapes might support the best and healthiest bees. If you think this is exciting and think it may be something you are interested in participating in, please send an email to dolezalbeelab@gmail.com so that we can add you to our growing list of citizen scientist beekeepers, and we will contact you when the website goes live, which should be sometime mid-March. I would like to thank you for watching this presentation and strongly encourage you and invite you to participate in this study and in this exciting and important new study as we work to help our favorite pollinators. If you have any questions, please feel free to reach out to any of us or visit our websites. Thanks again, and I hope you choose to participate in our study.